Okay, can somebody can hear me?
We can hear you.
Thank you. Let's give students a minute to log in. I hear they're still logging in. All right, before we begin, are there any questions? Not hearing anything. So today is February 6th, and we're supposed to be starting chapter 5, and that's where we are. Any questions about any of that? All right, chapter 5, microbial metabolism. This slide is showing you the major goals on the rough outline. Know the terms of this chapter. What's catabolism, anabolism? What is the activation energy? What is an enzyme? What's the active site of an enzyme? What is competitive inhibition? What's non-competitive or allosteric inhibition? What is a redox reaction? What are photoautotrophs? What are photoheterotrophs? What are chemoautotrophs? Chemoheterotrophs? What is fermentation? What is cyclic and non-cyclic photosynthesis? Any question about those terms? Remember, you need to know more than just these terms. You should learn the objectives for chapter 5. And for the objectives, you can find them under the class notes, for week 5 lecture, under the study guide. The objectives will be in the first part of the study guide for this chapter, and then the notes will be after the study guide, are the objectives. So there are the chapter 5 objectives, listing all the objectives you need to know for this chapter. And then there's the notes. It's similar to what we're discussing, only it's in sentence format and outline format, not a slideshow. Any question about any of that? And let's go ahead and make the notes to chapter 4 available, meaning the slide. All right. If there's no questions, let's go on with chapter 5. Uh, the second thing is understand what feedback inhibition is. And then the third and last thing, understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. Any questions about any of that? Let me blow this slide up. So microbial metabolism is all of the chemical reactions in a cell at one time. And for microbes, because most of them are uh, single celled, it would be all the chemical reactions in a cell or in the organism. You can divide metabolism into two parts, catabolism and anabolism. Catabolism is the decomposition reactions where you start with a large molecule, like large complex molecules such as starch, and break it down into a simpler molecule or molecules such as glucose. In the process, the energy stored in starch will be released because the energy in glucose and all the different glucoses will be less than all the energy stored in starch. Whenever you change energy from one form, such as starch, to glucose, you always lose some energy as heat. So this is just a second law of thermodynamics, and that is when you change one energy source to another, you always lose some of that energy as heat, meaning heat is always given off. some of that energy given off when you digest starch into glucose can be used to generate ATP. But realize that human cells are really lousy at doing that, meaning there's only two chemical reactions that human and generally animals can perform to make ATP. So there's only two of the catabolic reactions, and that is fermentation and aerobic respiration. The only two ways that human cells as well as animal cells can make ATP. In fact, green plants, which can make ATP in more ways than just two, I'm not sure how many ways they can do it, but oh, it's at least four, maybe eight, something like that. The point is that even in green plants, there are only a very few catabolic reactions that can make ATP. Most of the catabolic reactions do not make ATP. And so this slide is a little bit of an oversimplification. Any question about that? Such as when we're breaking, or even plants are breaking sucrose down into glucose and fructose, you cannot make ATP. And green plants can't either. Any question about any of that? All right. Catabolic reactions or decomposition reactions where you start with a larger molecule and you end up with a smaller molecule. In the process, energy is given off by the reaction. That's because the complex molecule has more stored energy than the simple molecules. Anabolism is the other half of metabolism, where you have a biosynthetic reaction where you start with simple molecules and you make larger, more complex molecules. You need to add energy to change a simple molecule into a larger, complex molecule. And that's because the simple molecules have less energy in them than the complex molecules. So anabolism is an energy using reaction. Usually in cells, not just human cells, but actually every cell I know of, when the cell needs energy, usually the cell obtains energy from ATP. It will break down ATP into ADP and inorganic phosphate. That's what the PI stands for. Come on, PI, inorganic phosphate. That reaction gives off energy. Some of that energy given off can be used by this reaction so that this reaction can occur, because this reaction cannot occur unless energy is added to the system. And usually that energy is added by breaking down ATP. So anabolic reactions are energy using reactions. Any question about any of that? No question about the heat being released? The second law of thermodynamics states that any form of energy, such as glucose, changed to another form of energy, such as starch, will give off heat. So some of that energy will be given off as heat. And heat is always given off whenever energy stored in one form is converted to another form. Any questions about any of that? All right. A metabolic pathway is a sequence of enzymatically catalyzed chemical reactions in a cell. So we can have one reaction, A, being made into molecule B, and enzyme 1 catabolizes or undergoes catalysis, meaning enzyme 1 makes this reaction occur. And then molecule B has another chemical reaction where it can be changed into molecule C, and that happens by enzyme 2. And then molecule C can be converted into molecule D by enzyme 3, etc., meaning D converted into E, E converted into F, each of them by their own separate enzyme. Oops, go back. You can string these different chemical reactions together, meaning A makes B, and then another enzyme takes B and makes C, C converted into D, D into E, E into F. This is called a metabolic pathway, where A will ultimately be made into an end product, which in this case is molecule F. The end product being the last product in the enzymatic pathway. Molecule B is the first product in the metabolic pathway, but it's also the starting substrate for this chemical reaction converting molecule B into C. So these products here in the middle are called intermediate products because B is the product of this reaction, but it is the starting substrate or reactant of this reaction. And that's why they're called intermediate products. The only last product is the only end product, and that's called the end product. And then molecule A is called the starting substrate or the starting reactant. Any question about any of that? Metabolic pathways are determined by enzymes in cells, and enzymes are encoded by genes. So this metabolic pathway would be encoded by the genes of the cell where this metabolic pathway is occurring. Any question about any of that? All right. There's a huge amount of metabolism occurring in the cell, and this slide is trying to show you all the chemical reactions going on in the cell that we know of. And we're going to talk about each chemical reaction. And you have to know each chemical reaction. Anyone object? That's a lot. We're not going to talk about that many. Chemical reactions occur according to the collision theory. The collision theory states that chemical reactions may occur when atoms, ions, or molecules collide with each other. When they collide, they can form the product, meaning the last step of the reaction, if they have enough energy for the reaction to proceed. And that energy is called the activation energy. The energy needed to disrupt the electronic configurations and the starting reactants to make the products. The reaction rate is the frequency of the collisions with enough energy to bring about a reaction. You can increase the reaction rate by increasing the abundance of enzymes. The more enzymes you have, the more likely the molecules that are the reactants will change into the products, at least more quickly. Another way you can increase it is increase the temperature. If you increase the temperature, the molecules are moving faster, jiggling more, and that means they have more energy in them. And so when they collide, they're more likely to have the activation energy they need to make the chemical reaction proceed. So increasing the temperature is pretty easy to see that it can increase the reaction rate of a reaction. Increasing the pressure does the same thing. And what happens is when you have a set volume and you increase the pressure, that means when the molecules kind of are closer together, but they have the same energy. And so they have, well, it's not exactly the same energy because the pressure is increased. They have more chance of becoming having the reaction occur, meaning there's greater chance they have the activation energy needed to disrupt the electronic configurations. And you sort of have to think of it as a volume, and then you compress the volume. The molecules are closer together, and so they're more likely to collide. And that's essentially why increasing the pressure um, speeds up the chemical reaction. Any question about any of that? All right, this slide is showing you a chemical reaction where the reactants will be changed into the products. This reactant actually has more energy in it than the products because it's a larger molecule, meaning A and B are connected. A and B as products are not connected. So it has higher energy, so you can have the y-axis, the amount of energy, and then this would be time. The reactant AB complex can break into the products A and B, giving off energy, which isn't shown here, but it is shown the energy difference between the reactant and the product. But to go from the reactant to the products, we have to go over an energy hump, meaning A and B, like sucrose, can be made into A plus B, meaning glucose or, let me see what it is, glucose or fructose. But to do that, you have to give the reactant energy, and that amount of energy required to make the products is called the activation energy. And there's the activation energy without the enzyme, and that would be quite a big energy hump. And then there's the activation energy with the enzyme. To get sucrose into, well, that's not a good example. Let's say to get glucose into carbon dioxide and water, we can do that. I don't know how much energy you need to give to get sucrose into glucose and sucrose and fructose. No, sucrose into glucose and fructose. I just don't know what it would be, but I do know how much energy you need to get glucose to change it into CO2 and water. And that is you need to heat it up to around 1,000 degrees. I don't know if that's Fahrenheit or Celsius. It doesn't matter. It's a very high temperature. You can take a blowtorch to the glucose and burn it, catching it on fire, and that'll produce CO2 and water. That energy does occur if I were to have a cup here, not really a cup, but you get the idea of glucose. Eventually, all of that glucose would be converted into CO2 and water. It's just that if I were to look at this tomorrow, it'd still be a cup of glucose. If I were to look at this in a year, it'd still be a cup of glucose. The difference is it's happening on the molecular level. And every once in a while, I don't know how often, one glucose molecule will be converted into CO2 and water. And obviously, if I look at the cup a year later, you're not going to notice any decrease, but there would be a slight, very slight decrease in the number of molecules of glucose. Any question about any of that? But you can add 1,000 degrees by taking a blowtorch to the glucose, and you'll very quickly convert it into all CO2 and water. And that is the activation energy without the enzyme, which is around 1,000 degrees, either Fahrenheit or Celsius. I don't remember. It's very hot, whatever it is. And then for evidence that energy is given off by this reaction, if I were to light the glucose right here, I would have to step back because so much energy would be given off by the glucose. If I were to catch it on fire by setting a blowtorch to it. Any question about any of that? And that energy difference between the reactant and the product is shown here. The energy is given off by this reaction. All right, but the activation energy with an enzyme is much less. You don't need to heat it to 1,000 degrees Fahrenheit. You only need to heat it to around 37 degrees, and then the enzyme can work converting glucose into CO2 and water. And that actually happens all the time in the room that you're in. Where is that happening? Where glucose is being converted into CO2 and water. Come on, somebody's got to state, because I'm not going to state this one. Should be obvious. I'll give you extra credit if somebody gets the right answer. Well, it's happening all the time inside your cells. They're at 37 degrees Celsius, which shows you that the activation energy, at least for converting glucose into CO2 and water, is much less with an enzyme than it is without an enzyme. There still is an energy hump. You do need a little bit of energy to make the products, but it's not that much compared to without the enzyme. Any question about any of that? All right. Enzymes are biological catalysts. They do speed up chemical reactions. You don't need to know how many, but it's a great amount, either 10 to the 8th or 10 to the 10th times. They speed up a chemical reaction that fast. And obviously you know that because we mentioned that glucose is going to be converted into CO2 and water. It just takes a very, very long time. And I don't know how long, but it's something like a century. So you have to have the cup out for a century before we'd notice water being formed in it. I'm not sure if you'd even notice because the water would obviously tend to evaporate, but if you had it sealed or something, you'd start to notice after a century. It might even after a century all convert to CO2 and water, but I don't think so. I think even after a century, you'd still have some glucose left. So it's happening all the time, just at a very slow rate. But every once in a while, glucose will convert into CO2 and water. And if I had my cup here, what would happen was a cosmic ray, that's a high energy particle from outer space, would hit a molecule of glucose, and then that glucose would be broken or given enough energy from the cosmic ray to convert it into CO2 and water. Uh, Enzymes are specific for a single chemical reaction. This is a little bit over an oversimplification. And as you know, there are some drugs that can mimic an enzyme. And so we give this drug so that the enzyme isn't converting the true reactant to the product because we don't want the product. We give the drug and then it will react with the enzyme tying it up. But generally speaking, most enzymes are pretty specific and for one molecule. Like I said, it's not entirely true, but it's very close to it. And so we're going to say that enzymes are very specific. They can perform one chemical reaction and essentially only work essentially on one molecule. So for each chemical reaction, you need a different enzyme. The specificity of an enzyme is due to its three-dimensional shape. And each enzyme acts specifically with its substrate by binding to the substrate with its active site. So there actually is a part of the enzyme which binds to the starting substrate. It forms a complex, and then the enzyme can perform the chemical reaction. And generally, the enzyme will only perform one specific chemical reaction, meaning to break sucrose into glucose and fructose, you need one enzyme, and then to convert glucose into CO2 and water, you need, actually in this case, you need several different enzymes, but we'll talk about that later. The point is you need a different enzyme. Enzymes are not used up by a chemical reaction. They're not altered permanently in a chemical reaction. And so when the reaction finishes, that enzyme is now free to bind to another reactant. It'll be the same molecule, just a different molecule. And then the enzyme can work on its second molecule. When the enzymes are working, like I said, they form an enzyme substrate complex where the enzyme actually binds to the substrate. The orientation of the enzyme and the substrate, and then the binding of the enzyme to the substrate, increases the probability of the reaction occurring. And what it does is it lowers the activation energy by putting stresses on the bonds of the reactant, so the reactant can more easily change to the products. Each enzyme works specifically on one specific substrate. And that's an oversimplification, but it's good enough for this class. Any question about any of that? All right. Enzymes normally end with ASE. ASE. So they usually end with ACE. And here's some reactions. Oxoreductase is an enzyme. Transferase is an enzyme. Isomerase is an enzyme. Ligase is an enzyme. I think we've already talked about catalase, an enzyme that can make changes hydrogen peroxide into water and CO2. Normally an enzyme, if it's named after they began this naming convention, I don't know if they made it in the 60s or 70s, probably maybe even earlier, but around then, so a long time. If the enzyme is named after the naming convention, it will end in ACE. The only enzymes that do not end in ACE are enzymes that were named before the naming convention came out. Like renin is an enzyme in a baby's stomach. It curdles the milk, so the milk will be a solid and then stay in the stomach for a longer period of time to be better digested than if the milk were to be uncurdled in a liquid, and then it would pass right through the stomach. I'm not sure how long, but it wouldn't stay in the stomach to be digested if it was a liquid milk. Any question about any of that? So enzyme names usually end in ACE. You don't need to know, well, most of these enzymes. We'll talk about ligase later. That's the only one we're going to talk about. So you don't need to know most of those enzymes. You should also know that enzymes do not work in a vacuum. We have some enzymes which require a helper, and that helper is either a coenzyme, it's a biological molecule, or it can require a cofactor, that is a non-biological molecule, usually a chemical element, something like iron, zinc, magnesium. And then the cofactor and the coenzymes actually bind to the starting enzyme to make the finished enzyme. So now that the let's see what that's called. Actually, it's here. The active site is now in a shape that it can bind specifically to the substrate. Any question about any of that? All right, this slide is showing you some important coenzymes. You do need to know the very first two, NAD plus or nicotinamide adenine dinucleotide. It's primarily involved in, well, really aerobic respiration, and we'll talk about aerobic respiration later in this lesson, which is why you should know NAD plus is an important coenzyme. Another is FAD. We'll talk about it with aerobic respiration as well, and it is an important enzyme in aerobic respiration. The others you don't need to know. NAD P plus is actually found in photosynthetic organisms. I probably should say coenzyme A because we're going to talk about coenzyme A later, but you don't need to know about coenzyme A, but you should know NAD plus and FAD are important coenzymes. This slide is showing you how an enzyme can work, and you can say an enzyme works by the lock and key model, meaning we have different enzymes which specifically bind to one substrate, much as one key fits to one lock. And how that enzyme works is the enzyme has an active site which physically binds to the substrate, so that's a complex where the enzyme is actually physically binding to the substrate. That puts stresses on the chemical bonds of the substrate to convert the substrate into the products, making it much easier to change the substrate into the products. Any question about the lock and key model of an enzyme? The next slide is showing you the hand in the glove model for how an enzyme works. And there's the substrate, there's the enzyme, and there's the substrate enzyme complex where the substrate is actually chemically bound to the enzyme. What do you notice about this slide? There's at least, well, there's three things, but there's two very easy to distinguish. So what do you notice about the hand in the glove model?
The substrate is inside the enzyme on the other one.
Yeah, the substrate will actually chemically bond to the active site of the enzyme, and that's being shown here, where the substrate moves over here, and then the enzyme changes its shape to almost surround the substrate. What else do you notice? Has to do with the size of things.
Enzyme looks bigger.
The enzyme is many times bigger than the substrate. Good guess. So yeah, that was what I was getting at. The other one, the third, you got to look really closely, and people probably won't even see it even. Some people won't see it even when I really blow it up. And here's the substrate, and there's the substrate there. You notice this substrate is flatter than that one.
So the substrate changes shape as well.
Yes, the substrate changes shape as well when the substrate is bound to the enzyme. It doesn't change its shape as much as the enzyme does. The enzyme really changes its shape. And so we could say this isn't the hand in the glove model. It's actually the shapeshifter's hand in the glove, where yes, the glove folds to fit the hand, but the shapeshifter hand folds or changes its shape to fit the glove. Those of you who have watched Star Trek Deep Space Nine, you know what I'm talking about with the shapeshifter. And the point is, the shapeshifter can change the shape of the fingers so that then the substrate is capable of binding to the enzyme. And then the enzyme is capable of changing shape and then binding to the substrate. And like I said, it's more than just the hand in the glove model. It's really the shapeshifter's hand in the glove model, where both the substrate and the enzyme are changing their shape. All right, any question about any of that? There are different factors which can influence enzyme activity. Enzymes have to work at a specific temperature, a specific pH, different specific salinity level. Generally, they have to work when heavy metals are not present, because heavy metals and alcohol and UV radiation can all damage the enzyme, causing the protein to denature. So my point is, enzymes do not work in a vacuum. They need to work under specific temperatures, under specific pH, under specific salinity levels, as well as the others, or else the enzyme will not work at its optimal level if it works at all. Any question about any of that? All right, another factor that can influence enzyme activity is the substrate concentration. If the substrate concentration is very low, you can increase the amount of enzyme activity, meaning how much activity the enzyme makes, by increasing the substrate concentration. Why? Because if you increase the substrate concentration, you're more likely to have a hit where the enzyme will bump into the reactant to form the products. So you're more likely to have that chemical reaction occur just by increasing the substrate concentration. You're also less likely to have the enzyme work, or you're going to decrease the amount of enzyme activity if you have poisons present. Poisons like cyanide and fluoride will damage the enzyme, and eventually, at least these two, will, once they've damaged the enzyme enough, that repair mechanism doesn't exist, and you damage the cell enough, you'll damage enough enzymes that the metabolic reactions in the cell cannot continue, and then the cell will die. And that's actually why cyanide does kill people. It binds to the enzyme, inactivates them, and then you don't get the chemical reaction occurring that you need. And that's how cyanide is a poison. Any question about any of that? All right. Two other factors you can have affecting enzyme activity is inhibition and then feedback inhibition. Inhibition can be in two types. There's competitive inhibition and non-competitive or allosteric inhibition. Whoop, it didn't work. There we go. I mentioned that an enzyme can be denatured. So here's the active three-dimensional shape of an enzyme under the correct salinity, temperature, pH, etc. And here's the totally denatured protein where the three-dimensional shape of the protein is totally changed. All enzymes have to work, at least at their optimal level, at their correct temperature, their correct pH. And if they're not at their correct temperature, it will decrease the amount of enzyme activity. So for our enzymes, they work best around 37 degrees. And any temperature below 35, that enzyme will not be working at its optimal level. It'll be working at a slower rate. And as you decrease the temperature, you'll get less and less of the energy, which can perform the chemical reaction until you totally freeze it, or in this case, until you reach 10 degrees Celsius. That's putting it in the refrigerator. And this enzyme cannot work at 10 degrees Celsius or lower, meaning that cell does not have much antifreeze, although it has a little, doesn't have much antifreeze. And so if you don't add antifreeze, it will, given enough time, eventually kill the cell. Any questions about any of that? The same thing happens with pH. This enzyme works best around pH 5. That's its optimal level. And then when you move away from pH 5, like pH 3 here, pH, well, closer to 7.2, 7.5 here, you'll note that the enzyme has less activity. And as you go down in pH to pH 2, or you go up in pH to pH, let's say 9, then more of the enzyme will be denatured. The enzyme will no longer have the proper shape. And if it doesn't have the proper shape, it can't bind to the substrate and then perform the chemical reaction. So yes, at a temperature, a certain temperature, as you lower it, you'll get less enzyme activity, but that doesn't mean the activity will be zero. The same thing with the amount of salt. If you were to put your item like in a bucket of pickles, there's usually enough salt in the pickles that it can pickle the great leaves just by heating them up in the microwave. Any question about any of that? Oh, let me state, but if you go very far away from the optimal temperature, the enzyme will be totally denatured and then it cannot function at all. So this enzyme isn't one that's partially denatured, it's totally denatured and should have no amount of activity with it. Another factor that can influence enzyme activity is the substrate concentration. If the enzyme is very low and the substrate is very low, then you get very little chemical reactions occurring, meaning you get very little end product. And the reason being is because the atoms, molecules, or ions that are going about, they're bumping into each other, but when they hit, they do not have the energy they need to convert the product into the molecules of the maize. Is that what I'm trying to say? Oh well. There's very few molecules together, both the enzyme and the substrate, and so as you increase the substrate concentration, you will quickly get more and more enzyme activity. That will eventually start to slow, and that's because the enzymes are now becoming bound to the substrate. And so if we increase the substrate further, we won't see much of an increase. And eventually, when we get the substrate concentration high enough, or you've waited with the chemical reaction for long enough, and then you do another experiment by adding more substrate, you'll see that the amount of enzyme activity doesn't increase at all. Why do you think that might be? Anyone have a guess? Take a guess if you want. I'll give you an extra credit point. The reason why the enzyme has no further activity as you increase the substrate concentration more and more, you don't see anything but a flat line in the enzyme activity. The reason being is at this point, meaning where the flat line is, all of the enzymes are now bound to the substrate. So it doesn't matter how much more substrate you give the person because there's no more substrate in the molecules. And that's why when you increase the substrate concentration, normally at the very low end, you'll see a great increase in the amount of activity, but that'll start to slow and eventually it'll stop. And that being because all of the substrate is already bound to the enzyme and you can't bind any more because most enzymes will only accept one molecule. So that's the only molecule binding to the enzyme. Any question about any of that? Another factor that can influence enzyme activity is competitive inhibition. The inhibitor competes for the enzyme for the active site. So for competitive inhibition, that's kind of odd. I haven't talked about inhibition yet, or did I talk about that last lecture? Did I mention about inhibition? No, I wouldn't have saved it, so this won't work. I was gonna look to see if I talked about that. Competitive inhibition is where the inhibitor competes with the substrate for the enzyme's active site. And so if you have enough of the inhibitor in there, it'll bind to the active site, tying up this enzyme, which cannot bind to the substrate, assuming the competitive inhibitor is in the substrate. And so that really changes the shape of the three-dimensional enzyme. But the real reason it's changed is because there's something bound to the enzymes, the yellow package, which alters the chemical reaction, usually by speeding it up. Competitive or allosteric inhibition is similar, except the non-competitive inhibitor does not bind to the active site the way that the active computer could. You could put it on hold and it wouldn't receive, well, text messages or phone messages. All right, non-competitive inhibitor binds to the enzyme at a site other than the active site. And we actually have a name for it. It's called the allosteric site. When the non-competitive inhibitor binds to the enzyme, non-competitive inhibitor binds at a site other than the active site. And when it binds, it changes the folding pattern of the protein by either breaking or reforming or forming chemical bonds that shouldn't be there. And as you change the shape of the enzyme, because the non-competitive inhibitor is binding, the active site could no longer bind to the reactant. And that's the way a non-competitive inhibitor works. It'll bind at an allosteric site, meaning a site other than the active site, changing the three-dimensional folding pattern of the protein, which then cannot bind to the substrate. No binding between the substrate and the enzyme, you get no chemical reactions occurring. Any question about any of that? All right, this is showing you feedback inhibition. Let me blow this up. We have our starting substrate, and it will bind to an enzyme here, which will then make the product here. Another enzyme can bind here, make water. Water in the third cell here can do its chemical reaction, making intermediate B. Did I move one too far? No, that's right. And then intermediate B can combine with enzyme 3 to make the end product of the chemical reaction. So this part is no normal chemical reaction. What's different is they have this end product, which can now bind to usually enzyme 1, but an enzyme very high in the metabolic process. When that non-competitive inhibitor binds, it'll bind at an allosteric site, meaning a site other than the active site, changing the folding pattern of this protein. So it's here, and then the active site can no longer bind to the penicillin B antibiotic, meaning the penicillin G. And that's the way feedback inhibition works. Here's a question for you. Usually in a cell, we see a feedback inhibition like this, where the end product binds to the very first enzyme. Why do you think that might be so common in cells? You want to have a guess? Actually, quite simple once you think about it. We're going to wait just a few minutes to try and get somebody to respond.
What was the question?
Why in a cell do you think we often see the end product binding to the first enzyme of a chemical reaction, meaning a pathway? Well, if the enzyme were to bind an enzyme, the end product were to bind an enzyme 2, what would we get, which you don't get if the end product binds to enzyme 1? All right, the reason why the end product binds to enzyme 1 is because if you shut down enzyme 1, you don't make intermediate A. And that takes energy for the cell to make intermediate A. So it's most energy efficient for the end product itself to bind to enzyme 1. Then you don't waste any energy. If, on the other hand, the end product were to bind to enzyme 2, you do make intermediate A. And the cell would waste the energy for making an intermediate A because it doesn't need it, because it has enough of the end product. And then the other reason why you frequently find a feedback inhibition like this, where the end product itself binds to the enzyme, and that is the end product itself is regulating itself. If there isn't enough end product in the cell, you won't have the enzyme, the end product binding to the enzyme and then shutting down this chemical reaction. You'll make more of the end product because you're keeping this chemical reaction going until you reach the point where there's enough end product in the cell, and then the end product will start to bind to enzyme 1, shutting down the chemical reaction, which shuts down the production of end product. Any question about any of that? So the answer was simply about the efficiency. The cell wants to be more efficient and not waste energy making a molecule it does not need, at least at that moment. All right. Before I move there, let me ask, are there any questions about factors influencing enzyme activity? If not, let's talk about ribozymes. Ribozymes are a non-protein enzyme. Most enzymes are made up of protein, and it's the protein that makes the enzyme. But there are a few enzymes which are not made up of protein. They are made up of RNA. And we have a name for that, ribozymes. All it means is it's an RNA enzyme. The RNA molecule itself can splice RNA. So RNA is acting as an enzyme. These ribozymes function as enzymes in that they have an active site that actually binds to the substrate. And then the ribozymes are also not used up or modified in a chemical reaction. So a ribozyme is an enzyme. It's just an enzyme that's made up of RNA and not protein. Very few enzymes are made up of RNA. Most of the enzymes in your cell are made up of protein, but not all of them. There's only one example of an enzyme that I'm even going to discuss, which is an RNA enzyme. And Tutora does not mention it, but he does mention the chemical reaction. And that is when you are making your protein, you first take the tRNA, bring the amino acid, the very first one, to the, let's just say the ribosome. And then you combine that amino acid to the second amino acid. What is removing the amino acid from the tRNA and then binding the starting protein, which in this case is just one amino acid, to the second amino acid, but it could be also the short amino acid. It's removed from the tRNA and then bound to the new amino acid that's just been brought in. That chemical reaction occurs from an RNA enzyme. It is the only RNA enzyme we'll ever discuss in this class. Any questions about any of that?
So basically what you said, the ribozymes, if I'm saying it right,
Yes.
are in the ribosomes where they join amino acids to form the protein chains.
No, that's one ribozyme.
Okay,
that
just one.
does that, just one. There's other
Okay.
ribozymes in the cell, and they are other RNA enzymes, and they perform a different chemical reaction.
Okay.
Okay. And I actually know of another one if you want to know, but for example, I've stated that they can cut and splice RNA. So other RNA enzymes can cut the RNA, others can splice the RNA together. So those are something that can do. Most of the RNA enzymes happen in a eukaryote cell inside the nucleus, but this one that I mentioned, where the RNA enzyme is removing the first or the earlier amino acids and then binding it to the newly added amino acid, that enzymatic activity is happening by an RNA enzyme. And I'm just stating that's the only one we'll ever talk about in this class. Tutora doesn't give you any examples for an RNA enzyme. He does talk about them, but he doesn't give you any examples. So remember, some enzymes, not very many, are made up of RNA. Most enzymes are made up of protein. All right, let's move on to metabolic diversity. There's metabolic diversity among all organisms, including microbes. all organisms, including microbes, require an energy source and a carbon source. So all microbes, as well as a cell, require an energy source where they obtain their energy, and then all cells require a carbon source where they obtain their carbon. You can classify organisms based on where they get their energy source. Organisms can be called a phototroph if the organisms derive its energy needs from light. So that's what a phototroph is. You can have a chemotroph, and these organisms derive the energy they need from a chemical compound. It may be an inorganic chemical compound, such as the chemosynthetic bacteria. We'll talk about them in just a minute. Or it can be an organic compound, such as glucose. Humans obtain their energy needs from an organic compound, such as glucose. So humans are chemotrophs. And everybody knows a green plant is a phototroph. You can also classify organisms on where they get their carbon from. There are two types, an autotroph, and they get their carbon from CO2, either dissolved in the air or in the water. Green plants are an autotroph, getting their carbon from CO2 in the air. And then people are heterotrophs. Heterotrophs obtain their carbon from an organic carbon source. And we get our carbon needs from eating glucose and another food molecule. So we are a heterotroph. Any question about any of that? You can classify organisms into both phototrophs and chemotrophs and then lump them together. We'll talk about that in a minute. It's more complex than just dividing an organism by its energy source into phototrophs and chemotrophs, and then dividing their carbon source into where they get the carbon from, either an autotroph or a heterotroph. So if ever you get confused, just take it back to this level and then figure out your answer. Any question about any of that? All right, so when we're talking about metabolic diversity among organisms, you can classify organisms into one of four categories: a photoautotroph, a photoheterotroph, a chemoautotroph, and a chemoheterotroph. This table is showing you the four different classifications for the troph. Let me blow that up a little. I think everybody knows that green plants are a photoautotroph because they get their energy needs from light. Everybody knows that. And almost everybody knows that green plants get their carbon from CO2 in the air. So plants are a photoautotroph. Let me state that more than plants are photoautotrophs. There is green or photosynthetic bacteria, which are photoautotroph as well, like cyanobacteria is a photoautotroph. And these are organisms which make oxygen, so you can call them oxygenic, meaning this photosynthesis in green plants and cyanobacteria can take light and CO2 and then make essentially glucose, obviously ATP or NADPH, and which can then be used to make the glucose. The point is they make oxygen. So cyanobacteria and plants are oxygenic photoautotrophs. You can have anoxygenic photoautotrophs. The green and the purple sulfur bacteria are photoautotrophs, but they do not make oxygen, so they're anoxygenic. I'm going to guess that nobody in class has ever heard of the green and purple sulfur bacteria. Is that correct? Anyone ever hear of them? The point being is you do not need to know the green and the purple sulfur bacteria. Any question about any of that? All right, the next easiest group to classify are the chemoheterotrophs, and that's because people are chemoheterotrophs. We get our chemical needs, we get our energy needs from a chemical, which is largely glucose or some other food product. And that's how we get our energy needs. And then we get our carbon needs also from an organic chemical compound, largely glucose. And yes, for chemoheterotrophs, oftentimes the molecule which supplies the energy is the same molecule that supplies the source of carbon. That isn't always the case. With some chemosynthetic bacteria, they will get their carbon source from glucose. So they break down glucose to get their carbon. But the chemosynthetic bacteria can use an organic chemical compound for their energy needs. What they do is they strip electrons from certain chemicals and then get their energy from that. And so this is very different. The chemosynthetic bacteria can be chemoheterotrophs, but so are we, and we get our energy needs from glucose. And that's where we get our carbon needs from too. So remember there's two types of chemoheterotrophs. Excuse me, didn't mean to do that. Those which get a chemical source from an organic compound such as glucose, and those which really are chemosynthetic bacteria, which get their energy needs from an inorganic chemical compound, such as H2S or sulfate, or they can get their energy needs from nitrite or from nitrate. Any question about any of that? Now, like I said, I've given an example of a organism that everyone knows is a chemoheterotroph, and that is animals and people. But also the fermentative bacteria are chemoheterotrophs. E. coli, when it's growing on a petri dish, is a chemoheterotroph because it can get its carbon source from the glucose in the media, and generally will use glucose as its energy source. But there are some bacteria which it's true they get their carbon from glucose or some organic chemical compound. So that's the same. But they can get their energy needs from an inorganic chemical compound. And it's only the chemosynthetic bacteria that can do this, as far as I know. The next group that's easiest to talk about are the chemoautotrophs. They get their energy source from a chemical compound. And in this case, it'll be an inorganic chemical compound like H2S or sulfate or nitrite or nitrate. And these chemosynthetic bacteria get their carbon source from CO2 dissolved in the air or the water. So note that the chemosynthetic bacteria can be either a chemoautotroph or a chemoheterotroph, depending on where they're getting their carbon from. They get it from CO2, the chemosynthetic bacteria is a chemoautotroph. If they get it from glucose, then that chemosynthetic bacteria is a chemoheterotroph. You probably don't know a chemoautotroph off the top of your head, but I'll guarantee you, you actually do know one. It's the iron oxidizing bacteria, which you guys have seen, I'll guarantee that, but you just didn't realize it. How many of you have taken up the back of the toilet tank, not the toilet bowl, but the tank, and you saw brown scudge or sludge on the back of the toilet tank, or it could be yellow sludge? How many of you have seen that? Maybe I'll ask you guys to raise a hand. Let me stop the share so I can see that. Raise your hand. That is a chemosynthetic bacteria growing in the toilet tank, and it's using chemicals in the water, largely iron, but it could be another chemical too, to obtain their energy needs. Because if you think about it, in that water, there's no food in there, no glucose. There's no sunlight getting in there. So it has to be a chemosynthetic bacteria. And they're chemoautotrophs. All right. Any questions about these three? The hardest for students to relate to are the photoheterotrophs. This is a organism, as far as I know, only bacteria, but an organism that gets its energy need from light. That's why they're a phototroph. And they get their carbon from an organic chemical compound such as glucose. The green and the purple non-sulfur bacteria. Before we talked about the green and the purple sulfur bacteria. Now we're talking about the green and the purple non-sulfur bacteria, are photoheterotrophs, getting their energy needs from light, but they get their carbon needs from an organic compound such as glucose. Any question about any of that? And I won't ask you for an example of a photoheterotroph because I don't think any of you have ever heard of the green and purple non-sulfur bacteria. I'm not even sure what they are. I've never worked with them. All right, let's go on. You do need to know in the classification system, you need to know the classification of all organisms shown in the red box. Let me make this into a slide. So we have all organisms. We can divide all organisms by where they get their energy source. If they get their energy source from light, they're a phototroph. If they get their energy source from a chemical, they're a chemotroph. You can then further divide the phototrophs into the photoheterotrophs. They get their carbon source from an organic chemical compound such as glucose. Or you can separate the phototrophs into the photoautotrophs, and they get their carbon needs from CO2. And as I stated, everyone knows that green plants are a photoautotroph. Let me remind you, you can separate out the photoautotrophs into the oxygenic photoautotrophs and the anoxygenic photoautotrophs. The oxygenic release oxygen in photosynthesis, and the anoxygenic do not release oxygen in photosynthesis. All right, the photoheterotrophs, you don't need to know the example of them because I'm sure no one has never heard of the green non-sulfur bacteria. Let's come back to the chemotrophs. You can divide them by where they get their carbon source. If they get their carbon from an organic chemical compound, they are a chemoheterotroph. And animals, fungi, protozoa, most bacteria are actually a chemoheterotroph. Not all, and I'm talking about the different species, because if you were to say the most bacteria, it's probably a, I'm not certain, but I'm going to guess it's a phototroph for sure, probably a photoautotroph. because there's a lot of different bacteria on the planet Earth, which are phototrophs. There might be fewer species, but there's more numbers in each species. All right, we can separate the chemotrophs into those that get their carbon source from CO2. These we call chemoautotrophs. And we didn't mention, you've seen one chemoautotroph, and that would be the chemosynthetic bacteria in your toilet tank. Chemoautotrophs. You can separate the chemoheterotrophs into what the final electron acceptor is. We'll talk about that later in this lesson. But the final electron acceptor can be oxygen, and people are that way. Most bacteria are that way. But there can be a final electron acceptor which is not oxygen. And something that grows in the absence of oxygen, meaning a obligate aerobe. If it engages in respiration, it will be aerobic respiration. Clostridium is an example of that. All right, any questions about that slide? If not, there's a good place to stop. So I'm going to let you off a little early. I'll see some of you in the lab at 6:30. The rest I'll see on Tuesday, I hope. Any questions? All right, I'll see you guys. Bye.
Thank you.
You're welcome.